All right, welcome back to another episode of the Exploring Health and Medicine podcast, where today I will be talking with Gabriel Baldanzi and Koen Dekkers, who are both researchers within the field of the gut microbiome. Now, the gut microbiome has been suggested to be involved in the development of various chronic diseases, such as cardiovascular diseases, such as high blood pressure, as well as metabolic diseases and chronic kidney disease. And so, depending on whether the gut microbiome plays a crucial role in the development of these diseases or a not so important role, which is something that I didn't know before I had this conversation with these researchers, I think that this topic is very relevant, or maybe not so much, but I didn't know before having this conversation. Now I know a bit more about the role of the gut microbiome in human health and in the development of chronic diseases. And we also covered the role of the gut microbiome in cognitive performance and in general well-being as well, and the role of the gut-brain axis in these effects that the gut microbiome may play. And so these are topics that I'm personally very interested in, and I did enjoy the conversation very much, and so I hope that you will be doing that too. Gabriel just defended his PhD thesis and has background in medicine. Koen has a little bit left before he is ready to defend his PhD thesis, but he's actively working on that. And so I present to you Gabriel Baldanzi and Koen Dekkers. Welcome, and I hope you enjoy the conversation. Okay, welcome Gabriel Baldanzi and Koen Dekkers to the Exploring Health and Medicine podcast.
Thank you. Pleasure to be here.
Yeah, for me too.
Right, and you both are gut microbiota researchers or microbiome researchers. That's correct, right?
Yes, that is correct.
But I don't think everyone knows this, but there's a difference between the microbiota and microbiome, right?
There are two terms that have different definitions. Although, when you read a lot of texts out there, even in the scientific literature, sometimes they are used interchangeably, which is not 100% correct. So both microbiome and microbiota refer to these microorganisms that live in our body. But when we talk about microbiome, we're talking more about the one definition is it would be their genome, so actually the DNA of these microorganisms. But microbiome has also been defined sometimes as like micro plus biome, and biome referring to the environment. So you could think it's microbiome, the microorganism, and the biome, the environment where they live. While when we talk about microbiota, then it's very specifically only about these microorganisms. So this living microorganisms that we have in our body.
Right.
Maybe I can add a little bit to that. So the microorganism in our body, we generally refer to organisms like bacteria, archaea, viruses, yeasts. So that's what we talk about when we say microbiota.
Oh, so it's not only bacteria. It's also yeasts and archaea and other microbes.
That's correct. And we work mostly in with the microbiota that live inside your gastrointestinal tract, so that we call the gut microbiota. And then it's mostly bacteria that we're looking at. But we also see some archaea, some yeasts. There are many viruses as well, mostly viruses that infect bacteria. But because they're so uncharacterized, it's definitely something that we don't focus too much on currently. But I think that's something that's very interesting for the future to look into as well.
Yeah. Do we know if any of these viruses that infect bacteria in our gut are impacting our health at all?
I don't think there's a lot of research on that yet. I think we are very in the beginning, on the first steps of studying this virus. There are phages, that's the name of this virus that infects bacteria. Of course, there are hints that they could, by influencing the composition of the bacteria, depending on what virus you have, they'll kill a specific bacteria and change the composition of the microbiome, and then consequently have consequences for health. But I think we're very in the beginning to understand if these viruses actually do impact the human health.
Yeah, I see. Yeah. But we do know that bacteria in the gut microbiota do, in some cases, definitely impact human health, right?
We do know that they produce a lot of substances, molecules, and that those molecules, they don't, especially the bacteria in our gut, these molecules don't just stay there. They're not inactive molecules. Some of them are actually taken up in our blood, in our body, in our bloodstream, and in our circulation, and could influence. And some of them, there is a lot of evidence showing that they do influence our immune response and our metabolism. So they're definitely, depending who is inside of us, that does change how our body is functioning.
And some of them are also opportunistic pathogens. That means that if you have a compromised immune system, they can be bad for you. But in normal circumstances, they're probably not so bad for you. Most of them have been living with us for thousands of years. So we sort of co-evolved together. So in general, they don't have a super big impact or a negative impact on your health. Most of them either have little impact, and maybe some of them might be a little beneficial even. They help with digestion of your food and produce compounds that your cells use. For example, I think an interesting example is dietary fibers. So those are different molecules found in plant-based food, and most of those dietary fibers we cannot digest ourselves. So there are some bacteria that can help us with that, and some of them produce what's called short-chain fatty acids. That's another group of metabolites, and those some of them there's quite a bit of evidence that it can be beneficial.
Right. I was thinking we were going to talk about that later, because there are some findings in this plasma metabolome large study that you've both been involved in, where you guys looked at the associations between various species as well as the total amount of diversity within the gut microbiota and various plasma metabolites, right?
Yes.
Right. So I'm thinking we'll go into the short chain fatty acid part eventually. But let's take a step back, and I want you guys to explain a bit about this study, how it was conducted, and what makes it so unique.
Yeah, we know that the microbiota, they produce a lot of metabolites, and some of them, they even enter our bloodstream, and that can have several different effects on your health. There's an example, and I'm still not sure if it's true. Each year there are papers providing different viewpoints. So there's a compound called TMAO, and it's actually it's derived from the microbiome. Then it's converted in the liver, and then it ends up in your blood. And if each year there are studies saying that it might affect your risk of getting a cardiovascular event, that it's associated with risk of cardiovascular disease. And I'm still not sure if that's true, but it's at least an interesting sort of direct connection between the microbiome and potential human disease. So, and the opposite is also true. We know that a lot of metabolites, they affect your microbiome. So there are generally sort of two big groups. So metabolites derived from your diet. So if you eat meat, there's a lot of metabolites that are in the meat that you eat, also in the plant-based food and all the other kinds of food that can affect your microbiome. And we have quite a few good examples of that. For example, metabolites that are found in coffee, they affect your microbiome. And another group of metabolites that has a very big impact on your gut microbiome, on the microbiota, is for medication. So medication, many of the medication that we take have very large effects on what kind of species and how many of them live in your gut. And we still don't know the impact of that, but we do know that it's true.
Yes. I'm very excited to dig deep into each of the topics that you mentioned. But first, can whoever of you feels like it, go into the methods of the study a bit more. So, for example, the metagenomics method. That's a method that was used in this study, right?
Yeah, so when we measure the microbiome, and Gabriel already said that with the microbiome, we often refer to as the genomes of the microbiota and the genes of the microbiota. And specifically, if we want to look at the gut microbiome, what we do is we would take a fecal sample, and then we would measure all the DNA that is in the fecal sample, and you can sort of get the DNA code by using DNA sequencing, and then as a sort of what you get is a lot of different short DNA fragments, and then we can look at those fragments and see which bacteria they come from, and in that way we can quantify how many of each bacteria and fungi and archaea you have in your gut. So this is what we often look at when we try to investigate the gut microbiome.
Yes. If I recall correctly, what the method that you used in this study to do what you just explained is called deep shotgun metagenomics. Is that correct?
Yes, it means that the shotgun means that we look at small fragments of DNA. So you can, when you sequence, generally you can look at short fragments, or you can look at long fragments. They both have advantages and disadvantages. Shotgun means generally looking at small fragments, and then deep is that you can sequence a sample for different amount of time, and there's a different cost to that. Deep means that you sequence it quite long, so that you get a lot of information, and that information is useful when you want to go looking at the function of what the different bacteria and other microbiota do, if you want to look at different strains of bacteria, for example, for those sort of more detailed investigations, you want to sequence the DNA deeply.
Yeah, I see. So that's really the strength of this study, that there was a large amount of genetic data that was analyzed. Is that correct? Is that a correct summary?
So when we started this this project, it was also quite a few years where this was feasible, this shotgun metagenomics method, but it was still quite expensive, and at that time many people still chose the, at that time, more sort of affordable and also more common method, which we call 16S sequencing. So that one works a little bit differently. Instead of looking at all the DNA in a fecal sample, you only look at a specific part of the DNA that's called the 16S DNA. And that has as the advantage that it's cheaper. It has as a disadvantage that you cannot look at the function of the microbiota, and you also cannot look really at the more detailed level. You will always have to be satisfied with looking at it at a little bit more broad perspective. So to be maybe more specific, with metagenomics, you can generally go to the species or sometimes even subspecies, while with 16S you are often limited to the genus level. So it's a more higher level of looking at the microbiota.
Right. And so, or did you want to?
I would just want to add one thing. Like, I think the genetic data, I mean, this microbiome data from this study was actually fantastic. But, I mean, this study, at the time it was published, I think what is also very nice and very unique and novel with this study was the number of people. Yeah. It's almost 9,000 people. It was not 9,000, it was 800 and something, or I'm sorry, 8,000 and something. And that was very unique by the time it was published. There was no study looking at gut microbiome with this quality of microbiome data and metabolomics or metabolites and plasma metabolites with this number of people. So that was also very fantastic.
Yeah. So a large number of people and also a more granular view of the genetic data.
Correct.
Yeah. But this also comes with a challenge, I think, because for me, trying to understand all of the data and all of the associations that you have published, for example, like as a reader who's not a researcher in this field, 298,982 significant positive and 247,837 significant negative associations between plasma metabolites and gut microbiome species, or even families or genus or so on. That's a bit hard to comprehend.
We thought the same. So that's a number that you really cannot discuss in detail if you want to write a story about it. So our sort of solution was to focus on a few aspects of these results that we found interesting. So we present them in the publication, and then we combine that with a website. And on that website, people can look up their favorite bacteria, their favorite metabolite, and see all the associations of that particular bacteria, particular metabolite. So that was our strategy. We also thought it was way too much results to highlight everything.
Yeah, yeah. And we've already mentioned some of these specific associations that you have gone into more depth about in your results and discussion section of this paper. For example, we mentioned short-chain fatty acids as a metabolite, or rather a group of metabolites that are influenced by the gut microbiome constitution. Would you like to explain a bit how short-chain fatty acids are involved in health and disease, and how gut bacteria influence the short-chain fatty acid metabolism in different ways?
Yeah, that is a great question. And it's one of those that maybe we could not assess not extensively with this study, specifically looking at plasma metabolites, because we know that a large part of these short-chain fatty acids, and let's name some of them maybe for some of the listeners, but it's like butyrate, acetate, propionate. A lot of them are actually consumed inside the gut. So especially, for instance, like butyrate, bacteria produce butyrate by the fermentation of fibers. But this butyrate, then it's used by the cells inside of our gut, our human cells, the colonocytes, as an energy source. So a lot of it doesn't even make into the blood circulation, where we measured all those metabolites. Yeah. But there are some good studies, some of them are my studies, showing that butyrate, by serving as an energy source to those colonocytes, they can perhaps also help gut to maintain its low permeability. And there's a lot of studies showing it could have positive effects on glucose metabolism and so on, glucose metabolism, but also avoiding some degree of inflammation by what some people call like the leaky gut, when there's a lot of content from bacteria getting from the gut inside the blood circulation. So having a more tight gut, not so permeable, could also be a good thing.
Would leaky colon be a more relevant term here?
Yeah, probably. Yes. Yeah. If we talk more specifically, yes, in the colon. Yes.
Okay. So, as I understand it, then butyrate is produced by different gut bacteria, and it is used as an energy substrate by epithelial cells lining the gut wall in the colon.
Exactly.
Right. And so, what species produce butyrate?
There's interesting. There are many species, and also they produce it by different pathways too. But I can name a few. So one it's kind of known out there is the Fecal prosnitzi. There are other ones like Bacteria rectali. What's the name of the other one? Streptococcus eutactus. But there are many species that actually can produce those short-chain fatty acids. But some of them are probably, let's say, more important because they also exist in higher amount inside the gut.
Okay. What factors might influence the amount of these bacteria that we have in our guts?
Diet probably affects a lot. Who lives inside of it depends what we eat.
Yeah, and what dietary factors might influence the butyrate producing bacteria in particular?
Well, that's what they eat, fiber.
Okay.
So higher fiber intake probably favors this bacteria. But also the type of medication that you use. So depending on certain medications we know have a higher impact on the gut microbiome, not just on these bacteria, but pretty much every bacteria that we have in our gut could be affected by some type of medication, antibiotics, for instance. And so And yeah, there is some evidence, and I think we'll come to that later, but there's also some evidence for physical activity changing the composition of our microbiome.
Yeah, we'll come to that. Yeah. Right. How about acetate or propionate? Are any of those? Because my interpretation of what you said about butyrates is that it's generally good to have butyrate-producing bacteria in your colon in particular, right? Are there any health benefits or maybe negative effects of bacteria that, or rather to have acetate or propionate in greater concentrations produced by gut bacteria?
Is that it less clear there? From all these short-chain fatty acids, the most abundant one is acetate. And I mean, the studies have been a bit not very consistent findings. I can say, for instance, acetate, some people found that it could, by putting acetate into the gut of mice, they could say some found an increase in blood pressure, for instance, but others did not. So it's not very clear. Maybe it's not just this short-chain fatty acids by themselves, but also the composition of the microbiome. So it's not well defined yet, the role of these other short-chain fatty acids.
Yeah, I think we like to talk about butyrate because we know a lot more about it. But I think definitely we will figure out more of that in the next few years, probably.
Yeah. Looking forward to follow that research. How much fiber? I know that you guys don't want to give like any specific recommendations. This is definitely what you should do in every instance, any medical recommendations. And that's not what I'm trying to ask. I'm trying to ask, like, what level are we talking about? Are we talking about milligrams of fibers that these bacteria will use to proliferate and become more dominant, and like that will have a butyrate increasing effect locally in the colon? Are we talking about milligrams? Are we talking about tens of grams? What level are we talking about?
I think an average fiber intake in the Swedish population will be between 20 and 30 grams of fiber per day. But if we know that most of it actually is not digested by us, it will end up in our gut or in our colon somehow and being consumed. Not everything is going to be consumed by bacteria, but a large part of it will be consumed by bacteria. But that's let's say, 30 grams of fiber is actually not very easy to achieve if you don't actually plan your diet around it. Okay. But most people will get close to 20 if they have a well-balanced diet.
Yeah.
Another aspect of the diet is it's not only the source of food, it's also affects how fast food travels through the gastrointestinal tract, and it slows that down quite a bit, and that might also be part of why we see health benefits for those diets that have relatively higher amount of fibers. So it could also be part of it.
Right. So on the one hand, I can see how that would, for example, improve glucose absorption rates, or rather decrease them to make glucose spikes less frequent. But on the other hand, isn't exercise something that generally increases? Well, not during the exercise, but after, like in the long term, doesn't it increase gastrointestinal motility and increase the speed of
Yes. It's even recommended for people who have problems with constipation, right, to exercise. Yes, that is probably what I said. We think that the gut microbiome composition could be affected by exercise. We don't know all the mechanisms, but as you mentioned, this could be one situation where by just changing the transit time, and that is how fast things move through our bowels, physical activity could be then influencing the composition of the microbiome. Now, if it's changing the bacteria who's there or changing how fibers get into the colon, that we don't really know the mechanism, but probably something in there too.
And these things, we don't know exactly what the mechanisms are that are beneficial or detrimental to your health. So often in biology, you get like, if you look at it from different angles, you see what seems to be maybe a contradiction, but the context really matters in most of those cases. So could be both healthy and unhealthy depending on the context. Right. We don't know yet.
We were talking about blood glucose, and of course when the blood glucose metabolism goes wrong, that is essentially a summary here, of course, but that is called diabetes, right? And so in this plasma metabolite gut microbiome study that you both were involved in, hydrocinamate and cinnamoyl glycine were positively associated with alpha diversity. Now, I guess I have two questions then. First, would one of you guys want to explain alpha diversity and why it is measured? And also, secondly, do you think that these associations have any clinical relevance?
Diversity, we often look at different sort of measures of diversity to look at sort of an overview of the microbiome. So say, in general, you have a lot of species, or you have many species in high abundance, and those are sort of aspects of the microbiome that have been associated with health-related traits before. So in general, we see that if you have many species in your gut, that's associated with traits that we would say are more healthy. Now, I do say it's associated. We don't know what is the cause and consequence, but we do see those associations. For example, with type 2 diabetes incidence, so that's why we often look at diversity as one of the aspects of the microbiome, not only at the specific bacteria, but as a sort of composite measure that says something about a healthy microbiome. Although I have to say that we don't really know if that's really a healthy microbiome.
I see. So if I understand you correctly, alpha diversity is a measure of how diverse the gut microbiome is?
Yes. So there are different ways to calculate that, and they look at slightly different aspects of what we would say diversity. But yeah, alpha diversity and beta diversity are sort of the two big microbiome aspects that look at how diverse your microbiome is, how many species, how they are distributed, how different your microbiome is from another person. This is a little bit technical, but this is sort of maybe the most understandable I can make it.
Well, we can make an analogy. Let's just think about a forest now in Sweden in December, where all the birds have flown away, and there's just some of them that actually can stand and still be in Sweden during winter. And you go to one of the forests and you try to find for the birds. Well, there's just a few species that are here. Most species have flown away. So the diversity of species in this forest is very low.
Yeah.
But if you come back to the same forest in July, when a lot of these birds came back, and those ones that were here in the winter, a lot of them stay until summer, then the diversity of species in the forest is much higher. So there are many more different species. So that's kind of what alpha diversity is in our gut. There's different types of birds or bacteria in our gut. That means a high alpha diversity.
Right. So it both encompasses the number of different species and the total number of bacteria, right?
Not amount.
Okay.
Not amount is more or less bacteria. But when we talk about abundance, it's how even the abundance is. It could be that you go to a forest and maybe now again winter, most of it is crows, and then there is one eagle or something, or one owl. Well, then most of it is crows, and I'd say then the alpha diversity is still low, even if there are a lot of crows.
Yeah.
But the diversity is low because even if there's an owl, it's just one. But let's say if you have half and half of it is or crows and half of it is owls, then your alpha diversity already has increased.
Yes.
Even if you still have two species, but it has increased because there are more in even numbers in this case. Wow, that is very, very simple example. In the microbiome, it's much more complex because we're talking about 400 or 500 different species, and then we want the abundance to be we don't want to have one species that is taken over. Because if you have just one species taking over, even if it's in a huge abundance, that means low alpha diversity.
Right, and that's generally associated. We don't know really like all of the implications of this statement and the causation and so on, but in general, lower alpha diversity is generally associated with worse health outcomes, right?
Yeah, so it's definitely associated. We don't know cause and consequence. Yeah.
Okay. So back to the hydrocinamate and cinnamyl glycine finding, which was that these metabolites were positively associated with alpha diversity, and they have previously been negatively associated with a lower risk of type 2 diabetes. How might gut bacteria influence these metabolites, and will it play a role in future treatments of type 2 diabetes, do you think? Is that possible that it could?
I have no idea. That's the honest answer. So if I remember correctly, these metabolites also make up, they're also found in your diet. So I have a feeling that maybe for these ones, it's actually your diet that affects your microbiome. And maybe that has something to do with it. Now, we also found with alpha diversity strong associations with metabolites that are produced by the microbiome. I think there was a steroid there with a very long name that I don't remember, and also picrosol. And picrosol we know is produced by microbiota in your gut, and it's quite interesting. It's a so-called uremic toxin. That means that it's a metabolite that in people with reduced kidney function, it's sort of accumulates in those people. So and because it's toxic, if you sort of get more of that toxic, it has negative consequences for your health. I think there with the alpha diversity associations, we really see a mixed bag. Some metabolites that come from diet, some metabolites that are produced from the microbiome, and I think it's fascinating to sort of try to figure out if what causes these people to get type 2 diabetes? Is it the diet? Is it the microbiome? Or maybe the people that have diabetes, maybe they have a different type of metabolism, and that's why they have more of these metabolites. So there are so many possibilities here, and that's why we are still working in this field.
Yeah. It seems like the field is generally almost like in the infant phase of its development.
It both is and isn't, right? It's quite an old field.
How old?
I think maybe 1600s or something.
Yeah, when we talk about the first studies looking at actually microbes that exist in our gut, then we go way back. But I think we never had really good methods until the last 5-10 years to actually look at these microbes with the with this refinement and resolution that we have nowadays.
Okay.
Yeah. So like I said, the method is probably new, not very new, but the methods that we are applying to this method, I think it's quite new.
Yeah. Do you think that this new methodology, like for example, deep shotgun metagenomics and such methods, are they as good as they ever will be for their specific purpose, or do you think that there will be entirely new methods that like come out and to a much greater extent?
Oh, definitely. So I talked a little bit about how you can look at short pieces of DNA, but also at long pieces of DNA. And here with the shotgun metagenomics, we look at the short pieces. However, currently the methods that look at longer pieces of DNA have improved so quickly the past few years that I see more and more people in the microbiome field thinking about, should we also try to look into that? And one of the big advantages to go to long pieces of DNA is that you can capture from one bacteria maybe the complete functional capacity. You can really see, okay, this bacteria has these genes, and it's all in one piece of DNA, so you're certain that it's all there. And then based on these genes, you can say, oh, this bacteria has this function and can produce this metabolite. So, of course, we know that if the current sort of rate of progress in methodology if it keeps improving and the cost will come down, that people will move towards the long read methods, the long DNA methods. But currently they're definitely a lot more expensive. And if you want to look at the number of people that we look at, like maybe thousands, tens thousands, then cost does matter.
Yeah. So the costs will come down. I get that, but will the most expensive methodology, or rather technologies that are used as methods in research currently, will they improve upon the current versions, or have we reached like a climax where this is it? Like we're at the final level of granularity of genetic sequencing of bacteria in the gut.
So just in the past few years, this method to look at large pieces of DNA has improved tremendously. Not only is it more affordable, but also the quality, the resolution that you get has improved tremendously. So I don't know, nobody knows if that rate will keep improving. But yeah, over the past few years, this really has been tremendous progress. So if it keeps improving like that, I think both when the cost come down and the quality of the data gets better, then of course people need to seriously consider looking at those methods.
I think one thing that will probably also improve a lot is how we do our sampling. I think there's a bit of a more distant future yet, but look, now we're looking at gut microbiome or gut microbiota by looking at fecal samples. So there's a large, let's say we're only looking at the last part of our intestine, or at least it's the overrepresentation of this. There's an overrepresentation of this last part of our intestine. But there are also methods being developed, and some of them are starting to be, tested and in the very advanced stage now, to have some kind of capsules that you can take this capsule, through your mouth, and as it goes down your gut, it will take samples. So now we can, in the hopefully not so far future, but I think it will take some time until this technology is completely available. But we're going to get also samples from your small intestines where we have very little idea of what the gut microbiome there is. So I think there is a lot of advances coming.
How does that technology work? Does it bring out like an arm to grab some fecal samples or other?
No, there are different capsules, but some of them is more like a passive absorption at a specific rate. Taking in this samples from within different parts of the gut.
So you mentioned earlier that there are a few hundred bacterial species in the human gut usually, like that's the average level. Do you think that with such technologies maybe we'll discover more species?
Yeah, probably. So we are somewhat limited by how detailed we can look at the microbiome, and that has already improved a lot in the past few years. But very rare species that are only found in very few people, we might not detect at a high enough level to pick it up. Also, another part of the equation is that we always compare our DNA that we measure against a database of DNA of bacteria that people have created. So we know this small piece of DNA belongs to this bacteria. Now, I think the methods will improve very much as well. So for bacteria, we have, I would say, the best databases, but for the other parts of the microbiota, the databases are a lot less good. So many of the DNA pieces that we measure, we cannot say it is from this archaea or it's from this yeast, because the databases are not as refined yet. So I think if in the future, when these databases get better and better and better, that also helps a lot.
Yeah. I think we can tie back to your question from the beginning about viruses, because there our database is very poor yet. I mean, compared to the database that we have for bacteria. So as we identify more viruses and create these databases, then we, of course, probably become much easier to study the virus that exists in our phage. It's virus and phages that exist in our gut. It's interesting also to highlight that we discovered new bacteria. It's not we as a group, but we as researchers, so other groups or other researchers. But new bacteria have been discovered by looking at human feces. So bacteria that we had no idea that existed before, we found them inside ourselves. And this is probably going to happen a lot with phages and viruses that exist inside our gut once we go more into them and create these databases and so on.
Yeah, interesting. Yeah. Sounds like there's still probably a lot to discover in this field.
Yeah, I think there's a lot to discover, and I think the methodologies will get better and help us with this large population-based studies. It's interesting. Genetics is not my good microbiome genetics, but human genetics is known as to be like a kind of successful field. And here the population studies with genetics, they are above 100,000 individuals. And now we have this study with 10,000 or almost 10,000 individuals, and we are like really large. Yeah. But we're not even close to getting the size that these human genetic studies have. Yeah. So when we get to those sizes, I think we will get much more data and much more interesting findings too.
Right. So it's on the one hand the amount of data, which depends on the quality of the methods and also the number of participants that you can gather data from. But then there are also the methods with which the data is analyzed, including artificial intelligence. And I wanted to ask you guys, how is AI used in the field that you work in, and how do you think that it may be used in the future?
I mean, everyone uses AIs like ChatGPT right now. That's true for all researchers as well, to a smaller and larger extent. And then regarding the methodology, I think there's probably a lot more to get there. I think people will start using this kind of artificial intelligence methods on all the data, not the microbiome, but all the data that we have. So they're currently focusing on maybe a different kind of things, but there are definitely some researchers out there that use these same methods already on the microbiome, and I expect there's not been super many publications yet with AI and the microbiome, but they will definitely come. I think it will be more common part of research than many people expect. Yeah. That's also hard to say. Nobody knows what the future will bring, but that's my expectation.
Right, I see. Other than using ChatGPT for like administrative tasks, which are a large part of the job as a researcher, as a person doing research, what other applications of artificial intelligence that maybe are specific to the field of gut microbiome research have you seen so far?
I think every time that we have large data, artificial intelligence, machine learning is something that can be used with good promises of results. And I think especially when it comes to prediction. So we have, like you said, we had like over 200,000 associations that study. We could not make sense to all of that. There's a website where people can access those results and try even help us to make sense of it. But because it takes so much time to look at those results manually. But if you have some kind of AI or machine learning that can look at those results and try to help us find patterns, so that could be helpful. But the other thing, also looking at the gut microbiome, and again, we have about 400 to 500 different species in our gut in one individual, but individuals have different microbiomes in different species. In total, there have been more than 2,000 species identified in the gut microbiome. We just have different amounts of them. But we can use that information, this over 2,000 different types of species in different amounts. Can we use all that data, all that information, and use together with some kind of AI machine learning methods to predict diseases, outcomes? I think it's a big discussion in this field if we can use machine learning here and do better than we can do already with the methods that we have. So, for instance, if you try to predict cardiovascular disease, what we use so far, age, blood pressure, total cholesterol, if you're smoking or not, if you have diabetes or not, those information gives us a lot of prediction already, right? Can we do better? Can we use gut microbiome together with that machine learning, AI, and help us in prediction? Of course, this is something that has to be updated all the time. because as treatment comes, medication comes, as lifestyle changes, these predictions will also change. Yeah. But and we have to recalculate all those predictions all the time. But if you have an AI that can do that better and faster, it would be really nice.
Yeah. I agree.
So there's so many people also working in drug development, right? Screening different compounds, and AI has had real progress there, and I think maybe the phages where we actually have so little information currently, maybe that is an avenue where AI can really make some immediate progress, because the databases are manually being curated and created right now, but maybe AI can be used there to help us.
Yeah. How did you select which? I'm guessing association strength and like effect size might be one method, but how did you choose which metabolites to focus on in your paper, and which associations to bring forth and have a deeper discussion about?
Yeah, so in general, we wanted to look as many metabolites that we could detect, because we don't know which one might be the most interesting with regards to the microbiome. So initially we started with as many as possible, but then we got so many results, right? And then we just started looking at the ones that for which we had the most evidence, and then we combined that with the metabolites and for which we knew something already, right? It's always when you find the metabolite that we don't know a lot about, then it's always difficult to discuss. It can be super important, but if we don't know anything about it, it can be very difficult to discuss. So we focused on the strongest findings and the ones for which we had additional information, what metabolic pathways they are involved. So that was our approach, and we discussed it with several people, and they found sort of several stories that they were interested in, and we decided to focus on those few, and hope that other people that have different favorite metabolites can use the website to find their own story.
That's right. So you guys focused on certain associations based on previous findings, and which metabolites have been associated previously with variations in the gut microbiome in general, and also with the particular species. But then you also created this Gutsy atlas, which is available for everyone, open source, right?
Yes.
Yeah. And I'll include a link in the show notes to that as well, so people can check it out. Yeah. As regards a particular metabolite that I think, or rather a group of metabolites, I guess, that people might be interested in, I am interested in it. I'm thinking about coffee and the metabolites that it does it increase these metabolites through including, like, as ingredients of coffee, being, for example, quinate is one I'm thinking about. Is that an ingredient of coffee, such as caffeine is, or is it something that is just increased in the human blood as an indirect effect of some event in the gut. Let's start with kinates. How does it, because it is increased in the plasma after consuming coffee, right?
Yes, so we have looked at several metabolites that were strongly associated with the microbiome, and then we found out that they are all part of compounds that are found in coffee. So the ones that we focus on are actually part of coffee, and of course, many of these metabolites also have get converted in your body to other metabolites, so that's definitely true. But the ones that we focused on here, I would say all, but maybe not all, but many of them are actually part of coffee itself.
Yeah, and so kinate is a coffee metabolite, which its variance in the human plasma is explained by gut microbiota to an extent which is greater than 25%, right? Can you help in interpretation of this finding?
Yeah, that's always difficult. I'm going to do my best. When you see that, for example, a certain a bacteria or diversity is associated with a metabolite, and in this case it's quinate, the coffee metabolite, you also want to know, okay, it's associated, but is that relevant, right? And one way to look at that is to see how much you can explain. So that means if I only knew the information of the gut microbiome, how much do I know of people, how much quinate they have in their plasma? So if I would measure your gut microbiome and the variance explained is around 30%, then you could use the information in your gut microbiome to know quite a bit about how much quinate that person has in his plasma on average, right? Not for each person, it's the same, but on average you could say, okay, this person is probably has relatively high quinate, and this person has probably relatively low quinate in his plasma. So that is what you could do if you calculate the variance explained. You look at the microbiome, and then you can say, okay, this person probably has quite a bit of quinate in his plasma.
Yeah, right. Quinate is an ingredient then in coffee, like caffeine, for example. Does quinate have any interesting biological effects that we know?
That I know.
I don't know.
No effect that I know.
Okay.
Yeah.
But so it's an example then, really, just of that coffee and its ingredients can be, well, the absorption of the different ingredients in coffee can be probably it can probably be modulated by gut microbiota. Is that correct?
I really don't know. I really don't know how these things work. We know that they actually. So the trick with coffee is, right? We really don't know if it's healthy or not. I think it's probably quite depend on the context. Every year there are probably thousands of studies that say it's good or bad for you. So it depends on how much you drink, when you drink, probably. But we do see that it's has an effect on your gut microbiome. and then I don't know the rest. I don't know if that's good for you, if that's bad for you. It is interesting though. Because we see that it changes your microbiome. What does that mean? So I think that's probably something that's worth investigating. Also have to be careful here. I say we see that it affects the microbiome. That is what I think is going on. It's not what we actually measure. Because it's part of your diet, I think that it affects your microbiome, but that's not actually what we measure.
Because you measured associations, not causations, right?
Exactly.
Yeah, causality. Yeah.
So actually, this kinase in your blood, it not necessarily reflect that your microbiome is interfering with how much coffee you absorb, but it could just be a marker of coffee consumption, and that we're actually looking at these individuals that consume more coffee have this microbiome, rather than saying, okay, it is the microbiome that is influencing how much you absorb of this nutrient.
Right. So it's not really clear if coffee influences your microbiome more than your microbiome influences your absorption of coffee.
Exactly.
Yeah, I see. But there has been one previously reported association between coffee metabolites as a group and the specific bacterial species Streptococcus salivarius, and you also found an association between this species and coffee, or rather, this was a novel association in your study, right?
Yeah, I think so. I think so. So that one is quite interesting. There is some evidence that suggests that many of the Streptococcus species, in certain circumstances, can be maybe or are at least associated with poor health, but this one is one that actually competing with some of these Streptococcus species that are considered or associated with bad health. So I think that might be an interesting sort of link to investigate as well. But yeah, that's all very tricky, and I think there in this particular study we find so many associations that there must be something really interesting in there that we just overlooked because we found so many things.
Yeah, I see. But so the hypothesis, which would probably need further research looking at this specific research question and investigating causality and so on, but the association here was that coffee metabolites were associated with an increased fecal concentration of the species Streptococcus salivarius, and the hypothesis that one might take from that for future research, if I understand correctly, is that maybe drinking coffee could protect against some streptococcal infections in the mouth, because this is where this bacteria usually lives. Is that correct?
It's definitely an interesting hypothesis to investigate, right? But for many of these findings, we could probably argue from multiple directions and find some supporting evidence that you could use to investigate the hypothesis from this direction and this direction. But what you just said is also something that I think is very interesting, at least, and it's. In your head, it might make sense that it worked like that, and most of those hypotheses that make sense, I think you could at least investigate it to some degree. That would be very interesting, right?
One group of chemicals that there weren't, or rather there were only a few associations with gut microbiota in the study was plasma nucleotides. Why do you think that is?
I don't know the details anymore about it, to be honest. So we also have to be sort of careful here. So we measure more than 1,000 different metabolites, but we don't measure all the metabolites that are there, right? We only measure the ones that we can detect and that we know. So we probably miss a lot of metabolites as well. So it's always you should probably not say that if you cannot find something, it's because it's not there. If you also don't measure everything that's there, so I would caution against that. But in our study, we don't find that many. I don't know why.
Yeah. So there, like I get your point. Like this isn't really conclusive evidence that there isn't any link at all between gut microbiota and plasma nucleotides.
It's definitely not an exhaustive study. Yep.
But so and also there are no clear mechanisms that might be relevant here for why plasma nucleotides might not be impacted.
I don't have a direct, immediate idea how that could be super interesting, but there might probably be some researchers that are very into those metabolites that have very good hypotheses about how that might be relevant.
I see. You were also both involved in a separate study, which was also in this more meta study, one could say, which is SCAPIS, which is the large study where all of the data was collected, and then this study that we're going to talk about now analyzed this data from a different angle than the plasma metabolome study that we just talked about. And this study then is about the associations between different levels of physical activity, sedentary behavior, moderate physical activity, and vigorous physical activity, and the diversity and the specific species of the gut microbiome. In general, how is gut microbiome diversity influenced by different levels of physical activity?
It's a really nice question. I just want to start by talking something about SCAPIS, because you mentioned it, and I think it has been a fantastic data source that we have here in Sweden, where we have this volunteer individuals coming from different sites or different cities in Sweden that have been part of this study, providing blood samples, fecal samples, and answering our questionnaires. And they wore this device. It's called an accelerometer. And that it's something that we also have in our cell phones, but it was a device they had over their hip during 7 days, and it measures body accelerations. And by measuring body acceleration, we can estimate and the time that they spend in this physical activities of different levels or even in sedentary behavior. It will be the lack of acceleration, almost lack of acceleration when you have in sedentary behavior. But what's interesting in this study is that, we found again this association. Again, because we talk about diversity being something often related to good health. And then again, we found evidence for that by looking at people that were more active or that spent more time in moderate or vigorous physical activity. They had a higher diversity of their microbiome. While those people that were more sedentary or spending more time in sedentary behavior was associated with a lower diversity, lower alpha diversity of their microbiome. So in a way, it's very exciting to see this replication of this finding that alpha diversity seems to be a trait of good health, not necessarily a cause of consequence, but it is a trait of good health. Also when we looked at physical activity. And even this is like when we took into consideration in our analysis of people's age, sex, some of their dietary components. We talked earlier about fiber, so we also took that into account in our analysis.
Right, I was going to ask that because we were talking about fiber earlier, and I think we talked about it in the context of butyrate, but it does also increase diversity of the gut microbiome, right? In general, like when you compare people who eat very little of dietary fiber with those who eat to the levels of the dietary recommendations of various countries, I guess.
Yeah, we do see that people that have a higher dietary fiber intake, they have a higher diversity too. Yes. And if someone could say, well, but those people that exercise more, they also eat more fiber, right? Yeah. That was something that we tried to take account in our analysis by using these mathematical models in the statistical analysis, where we call it adjusting. And that means we try to account for that factor. And even when we account for dietary intake or fiber intake, do we still see difference in the microbiome related to physical activity when we take out this influence of diet? And we do see that in this result, in this study.
Right. But we still cannot really say that, or can we? Maybe we can in this instance. Maybe there's more evidence in other, I don't know, I'm asking you. Yeah. Can we say, based on this association study or other research in the field, that there's a causal link between doing more exercise, more intense exercise? Is that always a good thing for improving or increasing rather the diversity of the gut microbiome?
Yeah, I think we're getting close to have a more definitive answer here, not just based on our study, but also looking at other studies that have been published out there. Of course, we're still like, we want more evidence to be sure. We want more studies, larger studies, better data to be sure. But we're just trying to get that information. I think we're not there yet to be sure, but a lot of things are pointing into that direction. Now, we also think of it, like physical activity, as you mentioned, is it always going to increase your alpha diversity? That, I think we're a bit further away from answering, because there might be a limit, of course, right? Even too much exercise could also have some harmful effect. So I don't think that we're going to find this linear association of, increasing one is always going to increase the other. But we do see, at least in this study, where we looked at physical activity in two levels, the moderate physical activity and also the vigorous physical activity, that we find this association with higher diversity, even for these two types of activities.
So vigorous physical activity increased, or rather was associated with an increase in alpha diversity to a greater extent than moderate physical activity.
Yes, and there's just where when in this analysis we accounted for the effect of each other, like for the we accounted for the effect of each other in this analysis too. So we're trying to look at them. Of course, there's limitations in these methods, in these mathematical models, but we try to look at them separately as two groups, vigorous and moderate physical activity.
Right, I see. Right, and so there was a part in this study which was accounting also for adiposity, and this did change some of the associations, right? Because the association between sedentary behavior and alpha diversity was no longer evident, but the both of the levels of physical activity continued to be positively associated with alpha diversity, right?
Yes, that is correct.
Why do you think that is?
Yeah, it's a very good question. And this is, well, first, why we did this analysis, right? Adjusting for adiposity. And to do that, we accounted for BMI and waist-hip ratio. So these are measures of adiposity. Of course, BMI could represent other things, but in the general population, we need to mention here that SCAPIS is a population of individuals with an age between 50 and 65 years old. So in this population, normally higher BMI will mean higher adiposity. There will be exceptions, but this is what applies for most people. But we always have this question is your for this population, is your physical activity affecting your adiposity, or your adiposity is actually hinder you from doing physical activity? So even there, where we think it's so obvious, physical activity makes you stronger, make you lose weight. But in the population, maybe those people that have a very high BMI, maybe that's what actually is hindering them from physical activity. Yeah. So that's why we kept that as a separate analysis. And we did that second model adjusting for this adiposity. Now, there is some evidence it's a different type of study. It's a Mendelian randomization study where you're looking at the genetic predisposition to higher BMI and also to your genetic predisposition to physical activity. And it's interesting that from your genetic predisposition for BMI is also associated with spending more time in sedentary behavior. Mm-hmm. And at the same time, your genetic predisposition to sedentary behavior is also associated with having a higher BMI. So the two seem to be affecting each other more than physical activity. Mm-hmm. So physical activity could be seen as this more separate type of trait that the effect there is more likely of physical activity influencing BMI than more of the opposite of your BMI affecting how much time you spend in physical activity. Mm-hmm. So this is one potential explanation that sedentary behavior and BMI are so related that in our analysis, when we also accounted for adiposity, we no longer observe that association of sedentary behavior and BMI, but we still see this association of physical activity and alpha diversity, even when we took into account adiposity.
Interesting. I understand. Could it be also that certain bacteria in the gut can, for example, increase plasma metabolites in certain ways that we'll come into that a bit later or we can take that now actually, as regards the effects of gut microbiota on catecholamine precursors. You guys found that there was both it's somewhere in this stack of paper, but I'm not gonna try to find it now, but there was one finding, which was that phenylalanine degradation was increased. I think it was from increased alpha diversity. Well, in that group, it was associated with that. Do you think that's correct?
I have to admit, I don't remember those details.
Do you think that maybe through catecholamine effects, maybe through other effects, could gut microbiota constitution influence the degree to which one enjoys physical exercise, and through that mechanism exert reverse causation leading to increased levels of exercise in people with certain microbiota patterns rather than exercise influencing the alpha diversity of people's gut microbiota?
I think it's definitely possible. There are being some mice study looking into that, where they say that by changing the composition of the microbiome of these mice were more willing to spend time in their will. So yeah, I think it is definitely possible. We're coming into something that's called the gut-brain axis. That is how your gut, the composition of your gut could influence your brain through different stimuli. So I think it's definitely possible, and there's a lot of interest in that, especially from the point of view of sports medicine, athlete performance. Could you improve athletes' performance by changing their gut microbiome? There's a lot of interest in that, of course. There's one really nice study when they found that in marathon runners, so people after running for a marathon, they have an increase in their gut microbiome, a certain specific bacteria that can consume lactate. Mm-hmm. So the hypothesis there was that you have these marathon runners during the marathon, lactate increase, some of this lactate get into the gut and favor certain bacteria. Yeah. At the same time, if these bacteria are in the gut and they're consuming lactate, that could help the runner to get rid of their lactate. Yeah. So there will be like an advantage for this runner for having this lactate in their gut. Yeah. I mean, this is a bit of a hypothesis. There's something, this increase has been observed, this increase of this bacteria that can consume this Veillonella, if you want to know the name. But this
what was the name again?
Veillonella.
Veillonella.
So this increase of this bacteria has been observed in these marathon runners. Now, if that really reflects in an advantage for those runners, that is a bit more unclear. The study did not look at the performance of these runners. But it's a very interesting mechanism, and then it talks in favor of the hypothesis you were mentioning now, that maybe, could be that the microbiome could help us be more active, or could, or increase our pleasure for physical activity. Or I even got a question the other day, could it just be that the bacteria in our gut, through this gut-brain axis, is just increasing our well-being, how we feel about ourselves? It's definitely possible. And then if you feel better, that you exercise more. So again, this is one study that we did. It is cross-sectional. That means that we're looking at the gut microbiome at the same time that we're looking at physical activity and comparing these individuals. We're not interfering. We're not changing a person's physical activity habits and seeing what happens with their gut microbiome, which I think would be a natural follow-up of this study.
And one mechanism by which the gut microbiota may influence mood through the gut-brain axis, I assume might be that sedentary behavior was associated in this study with a greater abundance of E. coli, and E. coli is one of the major producers of GABA, right? The molecule which is also a neurotransmitter and an inhibitory, the inhibitory neurotransmitter in the brain, the main one. Does this GABA that is produced by some of the bacteria in the gut, and in particular E. coli, does it cross into the brain over the blood-brain barrier?
Fantastic question. And it's a lot of people that, GABA, as you mentioned, it's well known to be this neurotransmitter in the brain. But what a lot of people don't know is that people working in this area of brain physiology, to know that most of the GABA actually is not produced in the brain. It's actually produced outside. It's produced in our gut. How much of it crosses the blood-brain barrier? Probably not much. And then how much get into that synapses, how much of it gets between the two neurons and can actually influence the brain? That's the question that we would like to know. And I don't think we have a good answer for that, but it's very intriguing, right? If you have these changes in GABA by your gut microbiome, could that actually get into your synapses in the middle of your brain and influence that? That is a great question that, it would be very interesting to know.
I think everyone wants to know if the microbiome really affects our behavior, right? Because they do produce many compounds that look like neurotransmitters, are neurotransmitters, hormones look like hormones. So to what degree does this actually affect us, our behavior? And I think we really don't know. But it's super intriguing.
And I think also important to highlight here that not everything that the gut produces or that the gut microbiota produce need to cross the blood-brain barrier to influence our brain. There are a lot of nerves, afferent nerves, like the vagal nerve, that is actually, connecting the brain to our gut, or actually the gut to our brain too. And just by influencing that nerve at the gut level, we can, by nerve impulses, change what the brain is feeling and sensing. So it doesn't necessarily need to cross the blood-brain barrier, everything that produces in the gut. It could actually just be using those nerves as pathway to connect the gut microbiome to the brain.
There's this new obesity medication, right? That pathway.
Are you talking about like Ozempic and other GLP-1?
Yes, Wegovy, Ozempic, those. those medications, they act on a certain pathway that affects our appetite. And there are metabolites that are produced by the microbiome that affect that same pathway. So there are some links there as well. Does that matter? We don't know. But there are many potential links to be investigated. I would say that not all those potential links might end up being true, but it would be so interesting if at least one of them would really be true.
Yeah. Do you think that fecal transplants might enhance mood?
Ooh, great question. I'm optimist to fecal microbiome transplant. Okay. I think that is something that we're going to learn more in the future about. We see these connections between the gut microbiome and so many health outcomes. Of course, it is quite complicated. There are so many other issues related to that, antimicrobial resistance and so on. But I think it's something that we need to study more. Yeah. Right? It's a great intervention if you want to know what happens when you change someone's microbiome. But of course, safety first here.
Yeah.
And I think we're currently sort of looking into these fecal transplantations. But I doubt a little bit that a lot of therapies will directly come from the fecal transplantations in the future. I think they're more about how your microbiome, if changing your microbiome, if that can lead to positive health outcomes. But the very tricky part about fecal transplantations is that you need good donors of good, healthy microbiome. And so what I think is very intriguing is what you see now is that people start focusing on what does happen with the fecal transplantation and then try to see what are the relevant species that have this positive effect. And then they try to come up with a sort of, it's called a consortium, which means it's several species that combined have this health effect, and then they try to make a pill of that, and then we can sort of, if we can cultivate these species outside of our guts, like let's say in a lab, and we can make a pill, then we no longer need donors, because I think that's going to be a very big issue if we want to scale this up to something relevant. But I do think there's so much potential there.
Yeah. What are the challenges to, for example, probiotics that include specific species in different concentrations for absorption and actual colonization of different parts of the GI canal? Like, can you design pills that are specific to colonize with specific species in a way that consistently increases the right species in the right parts of the GI canal to get the health effects that you're looking for, or is that an impossible task?
I think we're getting there. So obviously we only have, I think, 2 approved microbiome-based medications yet, and they're both coming from a fecal transplant. They're sort of fecal transplant-based. Okay. They're not a direct fecal transplant, but they're sort of, you take feces and then you do process it, and then you can use that as the therapy. But I do think what we used to have with maybe the probiotics that we used to take, the effect there very, I would say very there's not super much evidence, scientific evidence, that those work really well. And but the current sort of clinical trials that are ongoing, they really specifically look at, are these species able to colonize? Are they getting to the right body part, the right part of the intestine? And they do make great progress there. So I think maybe the old probiotics, we should maybe doubt whether they're super effective, but like the current clinical trials are doing something more different, where they specifically look at those aspects and see if they work well.
Yeah. What are some of the absorption and, I guess, colonization challenges, and how are they, like, what are the attempts to solve those? Like, can you outline some of the, like, what are the challenges to actually, from the pill to the guts and to persistence in the guts? Like, how do we achieve that?
It probably also depends a lot who is already living inside of you.
Yeah.
Right? Your initial gut microbiome, our microbiome has a characteristic that's called colonization resistance. And that is a fact that actually is supposed to protect us from pathogens. So what does that mean is, if a new bacteria comes into our microbiome, if we have a microbiome that is in a good balance, this new bacteria will have a hard time colonizing us, because everyone is living there harmonically and something new wants to get in. It's also interesting to point out that these medications that have been approved by the FDA as microbiome therapies, they are designed to treat or prevent Clostridium difficile infection, which is a well-known infection to come after an antibiotic treatment. But the reason why you initially have that infection from the beginning is because you probably had an antibiotic before, you reduce the diversity of your microbiome, and that this Clostridium difficile, this pathogen, found a niche or an empty niche to proliferate. So maybe at that time your microbiome is in a more susceptible or acceptance. There is a bit greater to take in this new pathogen, or this new bacteria from this therapy. So this situation where your microbiome has been devastated or has been affected by an antibiotic is one situation where you're actually more likely to take in new pathogens or bacteria from the exterior environment. It also changes throughout life. We know that children, they have an alpha diversity. That's actually, children, I mean here infants. They have a microbiota that has a lower diversity than us as an adults. And that's probably because their diet first because they're born sterile. And then after that, their diet is basically milk. So we also see that point in life is when you're more susceptible or open to taking new bacteria into your gut. Yeah. So who lives inside of us actually plays a big role, so who can stay. Yeah. Koen was talking about probiotics. There are some studies with probiotics that show that after 6 months after the probiotics, sometimes even less, only 10% of the people still have their probiotics in their gut. So most of the people, like 90% of the people, will just get rid of the probiotics after 6 months or so. Yeah. And who are these people that kept their microbiome or their probiotic? We don't know, but it's probably have to do with who was in the microbiome before the probiotic came in.
Makes sense, yeah. So would maybe the largest amount of benefit from taking bacteria in the form of probiotics in pill form would be if your alpha diversity is currently low?
I think that is a good hypothesis, and I'm not sure if it's good, but it's those people that are more likely to keep their probiotic in their gut when you have this microbiome more susceptible to or more willing to take in new members to their community.
And how do you guys think about then, because many of the probiotic supplements that are sold on the markets consist of sometimes just one, but sometimes like maybe 12 species. But then we mentioned earlier that there are several hundreds of species in the average gut, and in general, more diversity is good for your health. Will more harm be done than if you try to you're not trying to do that intentionally, but if you take a supplement which has large concentrations of a certain bacteria or a few number of bacterial species, does that have a risk of competing with the currently probably pretty good variety of species that are does that happen at all? Like, will there be a knockout effect in some people from taking probiotic supplements?
I don't think we have seen that. It's quite good because there are a lot of people out there taking probiotics, and I think we can say that they're probably quite safe. If they're good for every situation, we don't know, but they're safe. Okay. I think we have a lot of people taking it, and if there were any health issues, we probably would have already detected it. Yeah.
And some of them are also part of fermented food that we consume daily and in quite large quantities as well. So they're generally quite safe to consume, I would say.
Yeah. What foods or supplements do you personally consume? As regards, with the intention of having an effect on your gut microbiome.
I can say I don't take any probiotic. I might have taken it after I took an antibiotic for some reason, but more like any other person. And, maybe this will bring me some benefit without knowing if it's actually true. It's just a little hope there. But food, I would say the best that you can do is to have a very diverse diet. There are some interesting studies now that came out showing that, for instance, salad and those things are what brings more diversity to your microbiome. And it's just probably just food that salads and also fermented food. But there's two things here. Those are foods that we study a lot when it comes to microbiome, especially fermented food. There's a lot of interest in fermented food in the gut microbiome. And salads is because they have bacteria, right? It's not some other foods that do not contain as much bacteria.
And of course, is it the bacteria itself that has an effect, or is it because the fermented food has compounds that those bacteria have produced that have an effect, right? So I think we don't know all about that yet. I also don't take any probiotics or supplements.
I see. Yeah. And I guess fiber could be a relevant variable here as well with the salads and so on.
Yeah, it's a great question. Like, how do you get new bacteria in? Yeah. I think fibers, they get into, just the fiber itself, It gets more into the situation where we're trying to promote or favor certain bacteria that are already inside of us, but we're not taking new bacteria if you have some kind of fiber supplement that is almost sterile. So how do you get new ones inside? It's a different question than how do we promote those ones that are already inside of us.
Yeah. And so in general, you guys personally don't really do much as regards your diet for trying to alter your gut microbiome? Is that a correct understanding?
So my hypothesis is that if you have a quite diverse diet, you have also a quite diverse microbiome because they can digest different parts of your diet. My hypothesis that might be good for you, but I don't think we have any evidence that really concrete there. So we do know that diverse diet is in general quite good. Whether that is good because one of the reasons that it's good is because you also get a diverse microbiome, I don't think we know.
Yeah, I see.
The only two things that I started doing more after I started with the studies in gut microbiome was to exercise more and eat more diverse food and try to increase my fiber. But that I think everyone can do regardless of what their microbiome. It's probably good for you. It's not going to be bad for you. So.
Yeah, I think that's a good high-level takeaway as regards like what practical actions should people or should, I guess is a bad word to use here, but could give benefits to most people.
If it's mediated through the microbiome or not, I don't know, but it's probably good for you anyway. Yeah.
Right. There was another study as well, as part of the SCAPIS study as well. The OSA study is the one I'm talking about. So obstructive sleep apnea and associations with that condition and various aspects of the gut microbiome. So there were some measures used in that study of the consequences of obstructive sleep apnea, such as the AHI, ODI, and T90. Does anyone of you want to explain those for the listeners?
Yes, of course. Okay, I would just start saying, what is obstructive sleep apnea, for those that don't know? But it's a condition that is quite common, and here I'm talking about perhaps 20 to 30% of the population, of an adult population, older population, have this condition. And that is when during sleep, a person has an obstruction of their upper airway. And that's kind of like your tongue that falls and something and obstructs your throat, and then you cannot breathe.
Like the light form of this is snoring, right? But it's a very light form, yeah?
Exactly. And then, it's like when you're snoring, it's because your upper airway, where the pathway for your air to get from your nose and mouth into the lungs, it's a bit narrow, then you're snoring. But when it closes completely, then you have this obstructive sleep apnea. And then the person make efforts to breathe, but the air does not come in, and eventually the person will wake up or make a very strong effort to bring the air in. Okay, so that's obstructive sleep apnea. What was possible to do in SCAPIS is that some of these participants in SCAPIS, they took a device home. It's an instrument that they had a nasal cannula, so it was something monitoring air coming in and out of their nose, and they have something in their finger that was measuring the amount of oxygen that they have in their blood. So what happens when you have this obstruction of the airways is that your oxygen in the blood will fall. So you get what they call hypoxia. So these three measurements, and going back to them. So the first one, the AHI, that's apnea-hypopnea index. That's based on that nasal cannula. So based on the measurement of air coming in and out of your nose, there's an estimate of how many events of this obstruction of airflow and consequent apnea. Apnea means just not breathing. How many events you had of that type during 1 hour. Or it's a mean number of events per hour during sleep. That's the AHI, apnea-hypopnea index. The second one is the oxygen desaturation index, and that is related to oxygen desaturation. So that is when the amount of oxygen in the blood falls below a certain threshold. And again, that's the ODI, oxygen desaturation index, is the mean number of these desaturation events per hour of sleep. And the last one, that's the T90, and that's the percentage of time. So it's the amount of time of your sleep time that the oxygen saturation fell below 90%. Just so people have an idea, what we consider like a normal level of oxygen saturation is above 92. But most people will have that above 96, 97. So when we talk about above 90, that is already, we can say, a clear hypoxia.
Yeah, it's low when it's below 90, clearly. Yeah.
Yes.
Yeah. Right. And so the associations with different species and the diversity of the gut microbiome, that I would think would be intuitive is that if there is a greater extent of, more frequent and a greater extent of the hypoxias during sleep, then anaerobic bacteria would be favored. So those that don't require oxygen to live and proliferate would be more prevalent in the guts of these participants that have obstructive sleep apnea. Is that what was found in this study?
Yes and no. We found that too, but I'm very careful to talk about that. Other studies have also said that, we found a higher amount of anaerobic bacteria of the people that have more severe obstructive sleep apnea. But what we need to remember in the first place is that inside our colon, the last part of our gut, and your last part of our intestine, that is an environment that already is free of oxygen. So the amount of oxygen we have there is already very, very low. So bacteria that can survive there are those bacteria that already are what they call anaerobic bacteria. So bacteria that actually cannot live in cannot live in an environment that has too much oxygen, or they are what they call the facultative anaerobes, and that is the bacteria that can live in both types of environments with or without oxygen. So, I mean, it's hard to say that, we found that this species that were anaerobic were favored, let's say, or they were more common in people with obstructive sleep apnea, or it kind of just happens that they are anaerobic ones because those are just the ones that are more common inside the gut.
Okay.
But there are a lot of people, other studies that claim that. I'm a bit more careful here to say that we clearly see that those are the most anaerobic ones, or it's just because there are more of this anaerobic in the gut anyway. So it could be a bit of a coincidence.
Yeah. And hypoxia has a lot of effects on your health and on your metabolism as well. So it doesn't have to be directly related to anaerobic bacteria.
Yeah. So you mean that hypoxia influences human tissues as well, like the gut wall, for example, and might through that mechanism or those mechanisms influence.
Exactly. And if you have chronic hypoxia, it affects your health in multiple ways.
Yeah. And usually in the case of obstructive sleep apnea, in bad ways as regards cardiovascular and metabolic outcomes, right? Like in general, yeah. I was getting a bit of a different answer than I expected. I'm happy to be like challenged in my beliefs, but
it's a very common hypothesis, I would say, in people that first see the results, right? You see hypoxia, you see anaerobic bacteria, and you want to make that link. But because hypoxia is such a strong other effects as well, and all those might contribute to, for example, which microbiota you have in your gut. I think it's not the only hypothesis that is possible, and yeah.
So T90, you said, was the time spent in less than 90% oxygen saturation during the night while sleeping. And it was positively associated with gut microbiota species that have previously been described as involved in propionate production, which is one of the short-chain fatty acids that we mentioned earlier. Does this have any clinical implications?
I don't think anything that we could detect in this study to say that this hypoxia is favoring species that can produce propionate, a point that it could have clinical implications. So I don't think that is something that we can clearly state from this study. Of course, it could be. There are previous studies in mice studies that show that if you transplant microbiome from mice that have been into a hypoxia environment into mice that were not in hypoxia, so you're transferring, fecal microbiota transplantation from mice that were in hypoxia to control mice, the control mice will develop hypertension. I think, interestingly, obstructive sleep apnea in humans is clearly associated and is often seen as a potential cause of refractory hypertension. So there could be a possibility of sleep apnea changing microbiome and having some implications or changes into health. In this study, we tried to look a bit into that. We found one species that was associated with sleep apnea that we also found associated with hypertension.
So I was going to ask about that later, but we can go into that now. So that species is Collinsella aerophaciens.
Exactly.
Right?
Yes.
And it was associated with both higher oxygen desaturation index and also with higher systolic and diastolic blood pressure. But you were going to say something about that.
Now, yes, so that is one that, of course, this is not conclusive evidence. We're not saying that, okay, we know everything about it, that it causes, but I think this species is a very interesting candidate to take into follow-up studies, to continue understanding it more. Maybe we need to go back to the lab and transfer this bacteria into mice and to see what happens. But I think it's an interesting hypothesis here. Now I'm going to jump back to the propionate. Okay. So propionate production. So it is a short-chain fatty acid, but it has an interesting thing here. Now this is propionate production. This is the conversion of lactate into propionate. So lactate, as we talked before when we were talking about physicoactivity, and then certain bacteria are favored by lactate. Lactate is produced when you exercise, but it can also be produced in hypoxia. So now this is all hypothesis, but the hypothesis that we made from this study, since that we saw that people that had more severe sleep apnea, or more specifically more severe hypoxia related to sleep apnea, they had more of this.
More of lactate, because cells in our body produce lactate when they use anaerobic metabolism instead of aerobic.
Exactly. And that could perhaps favor bacteria that produce propionate using this lactate. It's all hypothesis at this moment, but we think it's an interesting question that would be nice to look more into.
I see. So it's as many of these topics that we've discussed today, very interesting on a mechanistic level to just understand how the human body works. But do you think that there could be any clinical implications of, if it is the case that propionate production is from lactate, which is increased from hypoxia?
I think we would need an extra information here that we don't have from this study. Is there anything related to this propionate production that is detrimental to health? And we don't have any evidence for that yet. It could be, even though we said this connection of sleep apnea, hypertension, potential mediation by the gut microbiome. But what component of the gut microbiome that could be mediating this association, we don't know. And it's probably, even if it is gut microbiome mediated, the microbiome is probably just one piece of this puzzle. Yeah. But if we can get more information into that, it would at least open or create information for future studies to try to develop some kind of microbiome target intervention. So if we could use the microbiome or interventions to target the microbiome, and that bring health benefits. but it's at this point we are creating hypotheses to follow up with the future studies and potentially find some targets for treatment.
I see. So possibly future research will have to tell.
Exactly.
So I was thinking we can talk a bit more about the effects of the diversity and maybe some specific species of the gut microbiome. Do you know if there have been any studies on the gut microbiome and cognitive performance?
There have been, but most of the studies, they are kind of, there are association studies in small populations. There have not been performed, because like the studies that we're talking about, that our group has performed, we are very fortunate to have very large data in like 9,000 individuals. These cognitive studies, they are in a much smaller population at the moment. So their results can be we don't know how much they apply to the general population.
I do think that we'll get studies like that in larger populations sooner rather than later. I would be really surprised if in the next 2-3 years we don't get a large microbiome study and associations with cognitive function.
Yeah, that sounds very interesting, I think. It's something I'm personally very interested in, like cognitive functions and how different biological variables influence cognitive performance. Are there any preliminary findings on this topic? Like, I hear that you say that the current evidence is quite weak and small, based on small sample sizes and so on. What does the current evidence say?
Most of the studies, they did find, this species or that species associated, I don't remember their names, but associated with better or worse cognitive performance. But some of the studies, they did not have this shotgun metagenomics technology that we have, or data that we have in the studies. They use what Koen mentioned before, that 16S sequencing. And then the resolution of the microbiome is not as good. So we'll say something about this genus of bacteria. But mechanistically, I think we lack a lot still in that field to know what kind of mechanism could link those two. Yeah.
And I think the few studies that have been performed also have quite conflicting findings, right? This one finds this species, that one finds that species. So I do think we really need larger studies for there to be a little bit more certainty about what these smaller studies have discovered, because they are not so consistent with each other.
I see. Yeah, and that's usually the case when there are small studies with imperfect measuring techniques and so on. That's often the case, that you get results in various directions, I think.
Yes, and of course they would have loved to have larger sample sizes as well. And you need studies that start small and explore new fields. And you see over time, when people find it relevant, that the larger studies will come. And if we just take the field of genetics, the largest studies now look at millions of individuals, and they still find new relevant things that were not discovered when they had 100,000 individuals. So it's often true that you can find more robust, reliable findings if you scale up, and we will do these kind of studies in the near future.
And just to mention something again about SCAPIS, I mentioned before all these individuals that have so many different biological samples, and they have answered so many questions. We have so many measurements, But cognitive measurement is also so interesting, but it takes so much time to answer a cognitive question. Sometimes you need someone to gather with this participant. So he's scaling up, having those cognitive measurements, the good ones, right? The good cognitive measurements, the questionnaires and tests in a large population. It's unfortunately tricky. It takes a lot of time, and we want to get so much information of these participants. It's hard to get everything.
And I think it's already amazing the type of evidence that, the type of measurements that were part of these studies, like the Accelerometer-based physical activity measurement, the OSA measurement. Those are really time-consuming and generally quite complicated measurements to do. And they were done in so many individuals, and that's what makes these studies possible. So, just like with cognitive function, it will be done, and I think rather sooner than later. But it is a massive undertaking to do these kind of measurements, and they involve many people, and participants need to be willing to do these kind of measurements. So, I think they will be done, but those kind of examinations are so time consuming and so tricky to do sometimes. So great data requires a lot of effort.
Yeah, that's true. We've talked about some ways to measure the gut microbiome through, for example, in the SCAPIS study and the different studies we've discussed. It was mainly through fecal samples and deep shotgun metagenomics, right? For the average Joe who is interested and is a bit nerdy and wants to know, what does my gut microbiome look like? Are there any tests on the market that are valid and reliable that can actually be trusted, or do you think people should wait until such tests are available in the future?
They are there. You can buy them. I don't know necessarily, except for the novelty and the personal interest in it, if it has any sort of immediate relevance to your situation in any aspect. Perhaps only the novelty and your being interested in is sometimes good enough. But don't do it if you want to look at your health, because we are really not there yet as a field. But if you have disposable income and you like it for its novelty and your own personal interest, I mean, you can do it. There are companies out there that provide microbiome information to you. Question the quality of the data. I know that currently we cannot really say a lot of things about what this has for effect on your health. But just for a novelty, if you have the disposable income, sure, go for it.
Yeah. I think the quality of the measurements of this, not all of them, but some of those those commercial kits, the quality is quite okay, quite good. But then how the interpretation of the results, I think, is the tricky part.
Yeah. There was this anecdote, right? There's a person who bought like many of these measurements or these kits and let the companies look at that person's microbiome, and they all gave different answers regarding to whether that person had a healthy microbiome. So maybe some of them were good and had high quality data and were correct in their interpretation. But who knows which one that was? Yeah. So they are currently, at least in this anecdotal, only one person. They all gave different interpretation of whether that person had a healthy microbiome or not. Also, I don't think we really truly have all the evidence that we need to do that particular interpretation to really say, based on this measurement, you have a healthy microbiome. I don't think we have all the evidence there. I think maybe we do have some of it, right? And maybe that's what they use, and maybe in some cases that is correct. But at least anecdotally, from this particular person who did it, I think I would personally not look at this particular avenue to see whether I have a healthy microbiome.
Okay, so do you think that there are any situations in which I should, as a future prescriber of antibiotics, be more conservative with my prescription or not prescribe antibiotics because, well, the indication may not be so strong and the benefits of the antibiotic may not be so, well, it might not save any lives. It might just, for example, reduce some duration of an infection by 1 day or something like that. But on the other hand, the negative effect might be that it disturbs the endogenous microbiome. Do you think that there are any such situations where I should consider the negative effects of antibiotics on the gut microbiome? Or should I just prescribe as much as is relevant for the positive effects of treating infections?
It's a very good question. We have been looking into antibiotics in the gut microbiome, and we do see that for certain antibiotics, the effects can last many years. How much does that have consequences for health? This is still something that we're looking more into, but do effects will stay with your patient for many years. But that said, of course, if antibiotics are needed, they're needed. Yeah. If you have an infection that really needs an antibiotic, then I think there's not much question about it, yeah. Especially if this infection carries risk for the patient or for the person. But I would say, and I think there's certain antibiotics, and it's shown, for instance, clindamycin has been associated with a lot of Clostridium difficile infection after an antibiotic. Fluoroquinolones have a large impact on the microbiome and also have a risk for certain other adverse events. So I think it also depends on the antibiotic. So I think those two information need to be weighted together. The risk of the infection together with what is the risk of what kind of antibiotic is being prescribed and what is the purpose. Is just to, as you mentioned, is it an infection if I don't treat has a lot of risk for the patient, or am I treating this infection because it might be good to reduce the time of the symptoms by 1 day, as when some certain upper airways infections where a lot of times if you don't do anything, the patient will get better anyway. You're just reducing the duration of the
exactly.
symptoms.
Yeah.
Then I think it's good to think what kind of antibiotic you're prescribing and maybe have to share that decision with the patient about it.
Of course, yeah.
I mean, if we know a little bit more, right, and it's possible that you could prescribe 2 types of antibiotics, and the only difference is that one has a longer lasting effect on your gut microbiome, it disrupts it longer, then that might be a good reason to prescribe one over the other, right? But I don't think we have conclusive evidence right now. But I can see that being a consideration in the future.
All right. Thank you very much, Gabriel and Koen, for coming on the podcast. I appreciate it very much.
It was very fun.
Thank you so much for the invitation.